Alright, we're on.
We're on air with it.
Alright, who is this person?
That's Harry. The basic principle.
No, who is this person? This is, yes, Gregory Mendel.
Oh. Should we write that down?
If you want. You don't need to know. I'll tell you what's important. I mean, if you want to know a little bit about Gregory Mendel, it's fine. He was an Austrian monk. He worked with plants. We'll talk about that a little bit later. He basically worked out some basic genetics. Because I think he may have mentioned to you that at the time, you know, it's talking about the 1800s, people were trying to figure out genetics. They had no idea about DNA or genes, and had none of that concept, and trying to figure out how's information being passed on. I said the big idea, I don't know if you remember this, is that things blended together. Now, there was a very famous scientist who was working at the same time Mendel was working, and he will be the feature of our next unit. And he didn't know about Mendel's work. Actually, Mendel actually sent him a copy, but he ignored it. And that was Darwin. When Darwin was working on his theory of evolution, one of the biggest problems with his theory was that he didn't understand genetics. Maybe he should have read Mendel's work, but he never did. So we'll talk about Darwin. Next unit, we're going to start with Mendel. But I'm going to mention something. What I'm going to talk to you guys about is like basic genetics. So in other words, it's going to be more complicated in real life than what I'm going to explain. Okay? There are going to be a lot of scenarios where the genetics is very simple. The one that you just did right now, very simple genetics. Which means it's very easy to explain it. Okay? But it's not always easy. Sometimes the genetics, or oftentimes, the genetics is quite complicated. So, take it with a grain of salt. I'm going to explain to you how you figure out, like, patterns of genetics and how you can predict what someone might look like. But it's, you know, in real life, it could be more complicated than what I'm explaining to you. So, let's start with this slide, okay? What do you see in this slide? A cat. Okay? Very cute. All right. Now, the cat on the left has a name, and its name is Cece.
Oh. Hey, that's
Now,
me.
email. You have the to button, you have the from, and you have CC. What does CC mean? Carbon copy. So in other words, it makes a copy and sends it to that person too, right? A carbon copy is exact replica. How do you make an exact replica of something? You have to clone it. So CC is a clone of her mother on the right. Now, exactly, they don't. That's the whole point of this slide, is I'm going to teach you genetics, and I'm going to teach you basic genetics, but it's more complicated. Because if it was so simple, then Cece should look just like her mother. But she doesn't. Can you give me a reason why she doesn't? Was that?
Two different genetics. Because like things together.
No, because Cece is an exact copy of her mother's DNA.
Yeah.
In other words, the DNA in Cece is the same as the DNA in this cat, yet they do not look the same. Now you might think, well, maybe her color will change over time. And from what I remember, in this case, the color did not change.
There's a glitch.
There's a glitch.
Are you laughing? It's not a reason.
I don't know, I was laughing.
So what happened?
What's the other thing that might determine outcome, other than genetics?
Nature? Environment?
Nature is genetics. Environment would be nurture. You ever hear that argument, nature versus nurture? So, what may be the explanation is that the environment in which Cece grew up is not the same environment in which the mother grew up. Because the environment does affect your genetics. Like, think about something like intelligence. I'm sure there are strong genetic components, but environment plays a role too. So, it's a often it's the interaction between the genes and the environment. Height. You might have the genetics to be really tall. But guess what would happen if as a child you were starved or you acquired some devastating disease? That could impact your height. So the environment is going to have an impact. But there is another factor that we never think about. We hear about nature versus nurture a lot. There's a third thing. So I'm going to ask you a really simple question. I want you guys to take a look at your forearms. Okay? Roll up your sleeves, look at your forearms. And can you guys see the veins in your forearms? Okay. I'm going to ask you 3 questions. I think I'm going to ask you 3 questions. First question. The pattern's the same. No, they're not. All right. That's the first question. The answer to the pattern is the same. The answer is no, they're not. Second question. If I took a cell from your left arm and I took a cell from your right arm and they did that whole DNA sequencing thing, would they be identical? Are you saying that genetics on your left arm is different than the genetics on your right arm? They should be what? They should be identical because what did you start off as? One cell that made a copy of itself over and over and over again, right? So any DNA on your left side should be the same as any DNA on your right side. There is one exception that you'll learn about in grade 12, but that's not happening here. Grade 12.
I know, but I don't.
Shh, quiet, Jessica. This is not a video about you. Environment. Did the left arm develop in a different environment than the right arm?
Yes, it developed a different part.
So in Francesca, the left arm developed in where? Where would your left arm have developed? It would have been mommy's womb. And where would your right arm have been? Mommy's intestine?
Womb, like.
No, it would have been the exact same what? Environment.
So like when you come out of the womb, there's 2 different environments. Like you could be in a doorway, deciding which room to go. This one's hot, this one's cold.
I think I need to hit the pause button at this point.
Hold up.
In other words, let's ignore that person. We like to have people say crazy things so we know what not to say. So the environment on your left side and right side are the same. So the genetics are the same. The environment is the same. Why are they different? Yep. No. It's not. I don't think it's that. She said you use one more often than the other.
Like one arm.
Yeah.
Didn't we agree it was identical?
It isn't. No, your vein patterns are not identical.
Oh.
They're different.
If your body isn't. How does it depend on the way you grow? You're dominating it.
Randomness. Is that what you're going to say? Oh. There's a randomness to all of this. Okay? And that's something that we don't ever talk about. But if you, I'll leave it to you tonight to look at this. Okay, we'll talk about it tomorrow. If you look up the Dion quintuplets.
The what?
The Dion quintuplets. It's a very famous case study where they took 5 identicals. They were actually 5 identical children. You know, we talk about identical twins. These 5 were identical. Yeah. And they basically took them from their mother and father and put them in a zoo and they raised them in the same environment because they wanted to see, well, what happens if you take individuals with the same genetics, same environment? What happens? I want you to look at it tonight. We'll talk about it tomorrow. Okay? Yes, Sabrina, it is highly unethical. This was done in Canada, by the way. Okay? The Dion quintuplets. I believe Dion is D-I-O-N-N-E. So we'll talk about that one tomorrow, okay? Alright, so this is what we're going to look at in genetics. We'll talk about basic inheritance patterns. That's what we'll start with, and then we'll come back to this other stuff, alright? So we're going to look at, recording here, we're going to look at basic Mendelian patterns. Quintuplets.
Quintuplets.
Okay, so now I know why people record these things at home when there's no one. All right, so we're going to go over some terminology because you need to know some terminology to understand this. Okay, so I'm going to start with just some really basic stuff. Okay, and what that means is you're going to paint a very simple picture of a characteristic. So give me an example of a characteristic. Hair color. Okay, so we're going to pick a characteristic. And that characteristic is going to be determined by only one gene. And that gene is only going to have two versions. And one version is going to be dominant and the other one is going to be recessive. So what we're going to look at first is something called a autosomal dominance pattern. Now, just some terminology, just so you understand this stuff. Did we talk about what a gene is? Well, that's how the gene is made up of DNA, but what does it actually do? What does it? Did we talk about what it encodes? It gives you a protein. I thought we talked about that. It gives a gene gives you a protein, right? And that protein does something. So the gene comes in versions. Okay, the allele refers to the gene version. So let me show you an example here, okay? So let's look it over here. Actually, this slide, sorry. So here we have two different types of genes. Okay? How do I know they're different types of genes? One has to do with what? Yeah, one has to do with length, and the other one has to do with color, okay? Those are two different genes because they deal with two different things, but they're also located in two different locations. So this is the gene for color. Now this is a chromosome, okay? This gene might be located over here. The address of the gene, where it's located, is called the loci, okay? It's right here, the locus is the genetic address. Okay, so you can see that the genetic address of the gene that gives color is different than the one that gives length. Because they're different genes. So they're located in different areas. Now, what do these two genes have in common though? These two right here. What do these two have in common? Yeah, what do they have in common? They both have to do with what? Color, right? So a gene that has different versions, those versions are called alleles. Okay? So this allele is a gene that says black. This allele is the same gene, but it says brown. Okay? So an allele is a version of a gene. If you look down here, we have a gene that gives length of fur. There are two alleles, two versions of the gene. One allele says long, and the other one says short. The genetics that you just looked at with the taster, there's one gene. Now you have to, part of your research is to look at the name of the gene. But it comes in two different versions. Two alleles. One allele says you can taste, the other one says you can't. Okay? That's very simple genetics. It doesn't get any easier than that. Okay. We'll talk about stuff that's way more complicated. Alright? But that's very, very simple genetics. Now, how many sets of DNA do you have? No, sets. Not chromosomes. Your DNA is organized into chromosomes. And I showed you the picture of what a chromosomal picture looks like, a karyotype. Showed you a picture of what a chromosomal picture looks like. Okay, that's kind of redundant. But how many sets of DNA do you have? You have two. One from? Okay. So, Mendel discovered something when he was looking at his pea plants, and He noticed certain patterns, and he came up with a couple of observations, and we'll talk about them. But one of the things that he noticed is that you've heard of meiosis, right? You ever done meiosis?
I know meiosis. No,
Meiosis is when you make sex cells. And there are two types of sex cells. There's an egg cell and there's a sperm cell, okay? So, during meiosis, the sex cells have how much DNA? One set, right? You have two sets of DNA, but what happens is in meiosis, they split. The law of segregation says that when you have a gene, and you have two copies of the gene, those two copies will split from each other, so that a sex cell only gets one of the two copies. So it's not going to get both. So here you have two chromosomes. So this could be gene. This could be the same gene. These are just alleles. So this could be, for example, that taster gene, where one allele says you can taste, the other one says you can't. And in meiosis, sex cell's only going to get one of those two. Okay, it's not going to get both. It's going to get only one of those two. That's called the law of segregation. Okay? So the alleles separate. Now, let me show you an example of this. Okay, so let's look at some examples, right? Because this is what Mendel was working on. He was looking at things like flower color, or maybe seed color or shape. Okay, he studied a lot of characteristics. So we're going to look at this one, height. What happens if you take a tall plant and you mix it with a short plant? Now, what do you get? You could get a medium plant. You could. But what he did was he took not just any tall plant, he took purebred tall plants. Now, how many of you have dogs? What's a purebred? Yeah, it's there's only the genetics of that type of. By the way, dogs are all the same species.
Oh yeah.
Right? Dogs are all the same. We'll talk about that when we get to evolution. But a purebred means it's not mixed, right? So a purebred tall means that if you looked at the genes for height, they would all the versions, all the alleles would say tall. Okay? Whereas if you look at a purebred short, and you looked at the genes for height, all the alleles would say short. Okay? Because it's purebred. Now, what happens if you mix the two? So we're going to look at something called a monohybrid cross. Okay, a monohybrid cross refers to looking at one characteristic, because mono means what? Mono means one. So we're going to look at one characteristic. So we're going to look at just heights. Okay? We're going to look at a monohybrid cross of one characteristic that's controlled by one gene, where there are two versions, two alleles. So here it is, height. It's controlled by one gene. One allele says tall, the other allele says short. This is a purebred. This is the other purebred. You'll notice that the letters that are being used, the letter is T. T for what? Tall. You'll notice that this letter is capitalized and this one is lowercase, because this is a very simple picture of what's called autosomal dominance. Okay? In autosomal dominance, one allele is dominant and the other one is recessive. That simply means that when you have a dominant allele, you see it. Okay? If an allele is dominant and you have it, you see it. If an allele is recessive, you don't see it unless it's partnered with another recessive allele. So if you have two recessive alleles, then you see it. So, what happened? What did Mendel notice when he put a purebred tall with a purebred short? The prediction would be you get something that is middle. Something that's in between. And you know what? Sometimes you do get that. But in this case, he didn't get that. What he got, all the plants were tall. All the baby plants from those two parents were all tall. So this is not blending. Something else is going on. Then he said, let's go one step further. Let's take all of these next generation plants that are all tall, and let's put them together. Okay? Let's put them together, and let's see what we get. And this is what he got. He got a particular ratio. Okay? And the ratio he got was 3:1. And you notice this a lot. Okay, you notice this a lot. So I'm going to explain to you how we got that 3:1 ratio. But I'm going to show you an example with, I think it's a guinea pig. I think it's a guinea pig. Alright. I think it's a guinea pig. Same idea. Now, Mendel did not work on guinea pigs, but it's the same logic here. This is a monohybrid cross, which means we're looking at one characteristic, and that would be what? What do you think the characteristic is here? Color. It's a simple picture, so the color is only controlled by one gene, and the gene has two versions. One allele says black, the other allele says brown. One of the guinea pigs is purebred black, which means both alleles are black. And the other one says purebred brown, which means both alleles are brown. Now, based on the way that these are labeled, black is capital B and brown is little b. Which one do you think is dominant? Black. So in this case, what happens when you get these two together? Well, the female that is black, because she's purebred black, can only donate what type of allele? Black. Does she have the brown allele? No. The male that has the brown allele can only donate what? Brown. Which means she will make sex cells that are only contain the allele black, and he will make sex cells that only contain the allele brown. Yep.
Ours doesn't match that. Ours is black.
Yeah, that's a different one. Yeah, that's another one. I'm going to come back to that one.
Oh, so we don't have that again.
You don't have this one? I didn't give you this one?
No. We're missing a lot of your signs.
Oh, really? Alright. Well, this is in school with you, so you guys. Okay, so what do you think the babies will look like? This is it. The babies are going to look like this. Babies are all going to be black. Okay? They're all going to have one allele for black and one allele for brown. Now, some more terminology. Homozygous is the same thing as purebred. Okay? If someone is homozygous, homo means same and hetero means different. If someone is homozygous for a gene, that means that the two alleles are exactly the same. So this is homozygous black, that one is homozygous brown. If someone is heterozygous for that gene, what do you think that means? So one allele is, in this case, one allele would be what? Black, and the other allele would be brown. But what's the color of the guinea pig? Why? It's dominant. So the definition of a dominant allele is that it always shows up. So because black is dominant and brown is recessive, the black always shows up. The only way you're going to see the brown is if it's paired up with another brown allele. Now, what happens in the next generation now? Imagine we have guinea pigs that are all heterozygous black, and we get them to breed. What do you get? Well, this is what you get. You go back to the law of segregation. That says that when you make sex cells, the two alleles separate. So, your guinea pigs are heterozygous. They have a big B, and they have a little b. When a guinea pig that's a female is heterozygous and she makes an egg cell, one of those two eggs will have big B and the other one will have little b. If your male is heterozygous and the male is also big B, little b, what happens when he makes a sex cell? Well, the sex cell can only have one of the two versions. So it's either going to have the big B or the little b. And it's 50-50. Think of it like a coin toss. What are the odds of getting the big B? Well, how many letters do you see? So it's 1 in 2. A 1 in 2 chance. Now you see this thing here? This is called Punnett Square. Okay, it's called a Punnett Square. And the idea behind the Punnett Square is very easy. If you look at one parent, so this parent over here, this is the female. Okay, that's the symbol for female. This is the symbol for male. It's not really important that you know that. Okay. The female is big B, little b. So, the way you do a Punnett Square is you set up columns and rows. Now, how many columns do you set up? Okay, well, how many alleles can the female donate? 2. She can donate the big B or the little b. So we need 2 columns, okay? How many alleles can the male donate? 2. So we need 2 rows. So what you do, this is called Punnett Square, you basically label this row, this column with, in this case, the big B, and this column with the little b. And this row is labeled with the big B, and this one is labeled with the little b. And what this says is that, well, in this box right here, if a female donates the big B, and the male donates the other big B, what do I get? I get a hamster that's purebred. Well, not purebred, sorry. A hamster that's homozygous. Homozygous black. But that's not the only part of that might happen, but maybe something else could happen. Maybe the mother donates the little b. Maybe the dad donates the big B. In that case, what's the genetic makeup of your child? No, genetic makeup. Heterozygous. Okay. What do we call the genetic makeup? As a name. Now, I didn't talk about it, but it's called the genotype. Okay? So it's called a genotype. Okay? The genotype is the genetic code. So, this is what? Homozygous or heterozygous? Yeah. Okay. Here's another possibility. Maybe the mother donates big B. Maybe the dad donates little b. In that case, you're going to have the exact same genetic code as this one. It's also going to be heterozygous. And it's also going to be what color? Black. Now, how do you get a brown guinea pig in this case? Well, mom has to donate little b. Dad has to donate little b. And what do you get? You get a homozygous brown guinea pig. The Punnett Square is just a way of setting up this chart that allows you to determine the odds, the probability of getting black versus brown. Okay? Doesn't mean that you're going to get this. You know, you could have 4 guinea pigs, they could all be brown. It's possible. It's unlikely, but it's possible. All this says is that if you have a guinea pig, that there's a 75% chance it will be what color? Why 75%? 2 out of 4. Does that mean that you're going to get 75% of your offspring will be black? It's just a guess. but it's a guess based on a mathematical model. Now we have intermission.